going to tell you a thing that will make you wish yourself here. The smallpox, so fatal and so general among us, is here entirely harmless. There's a set of old women who make it their business to perform the operation every autumn in the month of September, when the great heat is abated. People send to one another to know if any of their family has a mind to have the smallpox. They make parties for this purpose, and when they are met, the old woman comes with a nutshell full of the matter of the best sort of smallpox and asks what vein you please to have opened. She immediately rips open that you offer her with a large needle and puts into the vein as much matter as can lie upon the head of her needle, and after that binds up the little wound with a hollow bit of shell.
This excited remark was made by Lady Mary Wortley Montagu, the wife of the British ambassador to Ottoman Turkey, in a letter written on April 1st, 1717 to her friend in London. It aptly captures her awe and wonder at a strange practice she encountered in Constantinople, where people seemed to inject smallpox into themselves on purpose. This letter, written on April Fool's Day, might have been relegated to a commonplace prank between friends if not for the events that followed. It marked the beginning of humanity's consistent and relentless effort to protect itself from diseases which up to then had been largely regarded as the wrath of God, leading all the way into the year 2021, where vaccines, based on the same basic foundation, hold the promise of allowing humanity to return to a normal life. I am Nikhil, and together we shall traverse through the genius ideas and often lucky missteps which allowed humanity to forget diseases like smallpox and now hold out hope that life can again be normal in the 21st century. Welcome to the first ever episode of Pursuit of Prosperity, and in this episode we talk about vaccines. Before we get to what followed the letter written by Lady Montagu and look at the ideas and people that have brought us to where we are right now, it's probably worth taking a look at our past and seeing where all this started. First up, Shubham, a writer on the podcast. Do we have any idea where all this began?
While we have no idea when and where this really started, there are some historical accounts of variolation in the 16th century in China and in Bengal in India. Variolation is different from vaccination in that it's only related to smallpox. Other localized communities in many different parts of the world had also practiced it, but we have no idea where it really started. It was certainly not widespread in the Western world until the efforts of Lady Montagu.
That's unfortunate. It would have been nice to know the name or the story behind the discovery of variolation. Anyways, coming back to 1721. So what exactly did Lady Montagu do after she wrote that letter?
Well, she does quite a bit. Soon after she wrote that letter, she got a 5-year-old son variolated by a doctor in Constantinople. She then returns to England a year later in 1718. Then she does nothing actively for a while, but remember, she is an aristocrat, so she keeps company with the rich and powerful people of the English royalty, especially the Princess of Wales. It seems like Lady Montagu is lying in wait for the right opportunity. And in 1721, an opportunity presents itself as London suffers a smallpox outbreak. She wants to variolate her daughter and calls in a surgeon who is apprehensive at first, but then agrees to do it in the presence of some esteemed physicians of England, since this is the first time it is happening. The girl gets inoculated and she is kept under observation. Some rashes develop, but they soon disappear. She is perfectly healthy again. The word spreads rapidly. The Princess of Wales, who almost lost her child to smallpox, obviously wants to get her children variolated and gets permission from the king to set up a test.
So this is all happening in 1721, and I'm assuming the testing standards then were not something we would agree with today, right?
No, not at all. It is 1721. Enlightenment has not hit yet. In the first test, 6 death row prisoners are given the chance to be test subjects in exchange for a pardon. Now, funnily enough, one of the prisoners did not actually reveal he had smallpox as a child until after he got the pardon. All the variolated get a few days of fever and temporary rashes and then emerge healthy after. Now, to be sure that these people are immune to smallpox, a prisoner from the test, one Elizabeth Harrison, a 19-year-old young lady, is asked to take care of a young boy who has smallpox. They're locked in a room and asked to share the same bed. She emerges healthy even from this ordeal, but the variolation is targeted towards children, and all the subjects in the first test were adults. So now a second test is scheduled, this time with 12 volunteers, mainly children. 6 of them orphans for obvious reasons. All of them are perfectly healthy again after a few days. Now the Princess of Wales is in favor of variolation.
Two things strike me from your story. The first is that we were extremely lucky that not a single person actually fell sick during the trials, since there were high chances of that happening, and I cannot believe that the response would have been positive even if one person had fallen sick. Secondly, the Princess of Wales being in favor of variolation is something like a modern-day celebrity endorsement.
Yeah, definitely. It is hard to imagine that this would have won people over even if one person had fallen sick. People were generally skeptical of a medieval technique where you're injecting a foreign substance with a needle into your body. Who does that? It seemed like a mad endeavor. It could not have taken off without the celebrity endorsement that it got. All of this started as a passion for Lady Montagu to protect others against smallpox. With the celebrity endorsement of the Princess of Wales, a lot of people started talking about it. It went from a passion to a fashion. And like every fashion, there were the haves and the have-nots, the rich and the poor divide. People had started making money from variolation. It had become a family business for many. But there's one major problem here. Since live smallpox virus is being used, the chance of catching the disease is very high. If not done right, the inoculated person may get infected and also infect others. Many hacks ended up killing people by doing it wrongly.
These hacks are probably the 18th century version of modern-day homeopaths, but the existence of such a risk meant that there was space for improvement.
Yeah, to be sure, the variolation process was at best a high-risk, high-reward proposition. It could mean safety from a deadly disease or death itself. Something different needed to be done. A safer method had to be created.
Jonas Salk, the legendary inventor of the polio vaccine, when asked about its safety once said, "It is safe, and you cannot get safer than safe." The rapid journey from injecting people with actual smallpox, which carried a healthy risk of death, to the modern-day scenario where vaccines have become commonplace to the extent that a new one is a cause for celebration, is an astounding case study of how advances in science can snowball. This avalanche of scientific progress in vaccines begins with the father of immunology, Edward Jenner, doesn't it?
Yes and no.
This is Jishnu, a writer on the podcast.
The generally told story is that Edward Jenner was told by a milkmaid that she cannot get smallpox because she had had cowpox before, and this somehow led him to experiment with cowpox, and he came out with the first ever vaccine. But the truth is never so simple, is it? What this story does get wrong is the timeline and all the prior work that well preceded Jenner's publication.
So wait, are you telling me that people knew about cowpox and how getting it could protect one from smallpox before Jenner's work?
Yes, exactly. In fact, there are so many rumors and folktales from rural England and a few other parts of the world that milkmaids who had had cowpox aren't affected by smallpox throughout their life. In 1668, which is more than 50 years before Lady Montagu ever wrote her letter, a German doctor reported immunity against smallpox which was enjoyed by cowherds. A lot of people initially dismissed this as pure speculation and just a weird coincidence of sorts. But a few doctors did start experimenting with this. They first inoculated their subjects with cowpox, and when the disease subsided, they virilated them with smallpox. They observed that these people don't show the temporary symptoms of smallpox that normally virilated people do. And some of them were so confident that they did this on their own spouses and children. They didn't know why it worked exactly, but they were still so confident in the method. But no one had ever reported this publicly until Sir Edward Jenner came along.
How did Edward Jenner get to his revolutionary invention of the vaccine?
So we are in 1768 and Edward Jenner is 19 years old and he's doing an apprenticeship with a surgeon. A milkmaid comes to him and she says, "I'm immune to smallpox because I've already had cowpox before." And for the next 20 odd years, Jenner just sort of sits on this information and doesn't really do much about it. He just completes his studies, receives his MD, and just settles into life as a surgeon in rural England. Then all of a sudden, in the 1790s, he starts looking for subjects to test this cowpox-smallpox theory. But finding people who already had a minor disease like cowpox is extremely hard work. He understood that he had to inoculate someone with cowpox and then vaccinate them with smallpox. So Jenner decided to inject 8-year-old James Phipps, who was the son of his gardener, with fluid from the lesions of a milkmaid named Sarah, who had recently been affected with cowpox. And once James had recovered from cowpox, he vaccined him with smallpox, which then had no effect on him. In fact, he was so convinced that he took this single data point and tried to publish it in a journal, and he was laughed out of the room and rejected outright for obvious reasons. But this did not deter him, and next year he tried it with a few more people and he produced very similar results. And in 1798, he just decided to publish it privately in a work titled An Inquiry into the Causes and Effects of the Variolae Vaccinae. Interestingly, the word vaccine is drawn from the Latin word for cow vaccinae. While Jenner was specific about Variolae Vaccinae, which translates to cow pustules, Louis Pasteur later proposed that in honor of Jenner's work, this generally be called vaccine. Of course, there were a lot of criticisms of his technique, but it was largely welcomed by the general populace. So much so that in just 50 years, by 1853, Britain made it mandatory for infants to be vaccinated, and they even went as so far to impose fine or imprisonments on the parents who refused to do so.
It's amazing that it just took 50 years for a brand new technique to be so widely adopted. That aside, how did Jenner's technique actually work?
The basic idea behind how Jenner's technique, and really all traditional vaccines work, is quite simple. Our body has an immune system that fights off foreign matter, and when a harmful or foreign agent invades, it recognizes it and it tries to destroy it. It also has a mechanism to remember prior invaders and how it responds to them. So vaccines introduce our body to a weaker form of the same disease-causing agent. So your body can take time to fight this, since by nature it doesn't cause much harm. Once the body recognizes how to fight it and destroy it, it remembers it. And when the actual invader attacks, it doesn't need time to create a response. It is ready and it is immediately able to attack and destroy the invader.
So putting it in the context of smallpox, cowpox is a weaker form of smallpox, and after recovering from it, the body is able to fight off smallpox when it actually invades. But cowpox is a naturally occurring virus. When did we start creating vaccines in a lab and start creating new vaccines for other diseases?
That's right, cowpox is naturally occurring. And this approach of finding a weaker natural pathogen for every single disease out there is simply not feasible. Creating vaccines in a lab was the next step in vaccine development. And it happened only almost 100 years after Jenner's publication with the work of the famous French scientist Louis Pasteur. But I'll let Arnab tell you more about it.
So, if you drink your milk directly out of a pouch or a carton, you should probably thank Pasteur for it.
Meet Arnab, a writer on the podcast.
So milk has a very short shelf life. If you don't process it within a couple of hours, it goes bad. Pasteur was working in the field of microbiology and he realized that there were microbes in the milk which made it to go bad. He then came up with this technique which helped us kill the microbes and store the milk for longer. To honor him, that technique was named after him. It's known as pasteurization. So if you carefully look on the labels of your milk pack, you'll see that it says that it is pasteurized milk. But Pasteur's contribution to vaccines was kind of an accident. Back in 1881, Pasteur was working on chicken cholera. That year during the summer, he decided to go on a vacation. While going away, he left a culture of chicken cholera on his desk itself, and once he was back from his vacations, he resumed his work. He wanted to inoculate some chicken, so he used that old culture that he had left on his table itself. Somehow, even after the inoculations, the chicken didn't fall ill. So he prepared another fresh culture for inoculations, believing that the old culture must have gone bad. Once again, he inoculated the chicken, but this time with the new culture. To his surprise, the chicken didn't fall ill again. Now this was something unusual, and Pasteur went into retrospection. He developed an hypothesis that it might be possible that the first inoculations with the old culture that was left out through the summer had done something to the chicken. It might have made them immune to the disease. So whatever Pasteur did accidentally is today known as attenuation. It's just a process by which we expose the pathogens to harsh environmental conditions to make them inactive.
That's quite a discovery. Probably a lesson to everyone that they should start taking more vacations to spark new ideas. But what happened next? How did vaccine technology evolve after this?
Now, Pasteur had stumbled upon a very interesting discovery, and he wanted to test out his hypothesis for the cause of these unusual events. He started working on anthrax and repeated the same process that he did earlier. It turned out that his hypothesis was correct, and he continued his work to produce the first vaccine for anthrax in the same year. He then took up rabies and produced a vaccine for it in 1885. Now this discovery of attenuation opened the floodgates of vaccine development. Soon there were independent teams working in Europe and United States who were using Pasteur's techniques to develop vaccines for other diseases. Soon they were able to develop vaccines for typhoid, cholera and plague. Now back in the time, this was a huge progress. Even though these diseases aren't serious today, but in those times they used to kill thousands. Following this, a large number of vaccines were created. By the 1960s, we had vaccines for TB, polio, tetanus and diphtheria.
At this stage, we have techniques to attenuate and techniques to inactivate harmful agents. Following this, I guess we play God and re-engineer the genetic code.
Exactly. So one thing that I should explain before we go ahead is the concept of an antigen. Anything that causes our immune system to respond is an antigen. This is usually a part of a microbe or a foreign object that is injected into our body. Fast forward to the 1970s, and now we are in the era of genetic engineering. We are now able to add or remove certain genes from a microbe, and this allowed us to come up with even better vaccines. We started working on a class of vaccines known as recombinant vaccines, which are products of genetic engineering. In recombinant vaccines, we take a harmless agent such as yeast and reprogram it to produce the antigens of a harmful pathogen. Using this technique, we developed the first recombinant vaccine for hepatitis B in 1986. But more importantly, this technique allowed us to develop vaccines without injecting harmful pathogens into humans. We could simply produce the antigen and directly inject it into the recipient. This also meant that we wouldn't have to handle toxic components and live pathogens anymore. This made the vaccines more efficient, safe, cheap, and pure.
It's astonishing to think of the fact that just 300 years ago, the first human trials for smallpox radiolation was being conducted using the live virus. Now, we are not only avoiding using the live pathogen, we are also capable of changing a microorganism's genetic code. If this episode was recorded in January 2020, this is possibly where the episode would stop. Modern-day vaccines are mostly based on recombinant DNA technology, and there are a large number of vaccines under trial which promise to make humanity immune to an even larger number of diseases. But it's not January 2020. The last 18 months have been an absolute roller coaster. Millions have lost their life to a strange new virus called SARS-CoV-2. But as we hopefully look at the tail end of the pandemic, a new technology has emerged in the world of vaccines, one that could revolutionize the pace of vaccine development and delivery. In late 2019, there were troubling reports from Wuhan about a scary, transmissible pneumonia-like virus. It was spreading fast, traveling across borders at breakneck speed. On January 10, 2020, Professor Zhang Wangjin released the complete genome sequence of SARS-CoV-2 online through a colleague in Australia. The virus was moving quickly, but information in the age of the internet was faster. Researchers at Moderna, a biotech firm specializing in mRNA in Cambridge, got the readout. Their scientists were able to design a vaccine on paper in 48 hours, and inside of 6 weeks, they had chilled doses ready for tests in animals. The question that stands to be asked, Jishnu, is how is it possible to design and produce a vaccine so quickly, and what is this mRNA technology?
Before we get into mRNA, let's look at some basics, yeah? Proteins are the building blocks of human cells, and these cells constantly produce proteins based on the genetic information which is present in their DNA. The DNA is present in the nucleus of the cell, whereas the protein synthesis occurs outside, in the liquid that fills the cell called the cytoplasm. mRNA or messenger RNA is an RNA molecule that contains genetic information and carries it from the DNA to the cytoplasm, where the corresponding protein is being synthesized. And hence it got the name messenger because it simply carries information from one place to another. mRNA vaccines contain strands that have the genetic information to make the viral antigen. The antigen is hence synthesized inside the body. And from there on, our immune system just does its thing, as with conventional vaccines, and creates the required antibodies. Importantly, the mRNA molecule is transient, and the enzymes quickly degrade the mRNA as soon as the protein is made. Since the mRNA technology doesn't need the virus itself to create a vaccine, and just needs the viral genome sequence, researchers were able to design a vaccine within days after they had the genetic information.
Absolutely fascinating. For how long has the mRNA tech been in existence, and why have we not been using it to develop more and more vaccines till now?
So the basic idea is that you create an mRNA strand that codes for a specific antigen. You inject it and the body creates that antigen and the immune system simply takes over. It sounds simple enough, doesn't it? But what sounds simple in theory proved much harder in practice. The first direct gene transfer in mice was achieved as early as 1990. But what we fail to realize is synthetic RNA is a foreign object to the body, and the body's natural immune response was heavy against it. This made the mice sick, they lost weight, and they stopped running around, and given a large enough dose, they even died within hours. Because of these adverse side effects, academia largely gave up on mRNA technology in the coming decade. But the relentless efforts of two researchers from the University of Pennsylvania, Kathleen Carico and Drew Weissman, proved ultimately fruitful. Kathleen Carico was in fact demoted in 1995 after 6 unsuccessful years of research in mRNA, but she was persistent in her faith in the technology's potential. After a decade of trial and error, she and Drew Weissman achieved the breakthrough in the early 2000s. The scientists determined that they could avoid the body's natural reaction to synthetic RNA altogether by using chemically modified building blocks to create a hybrid mRNA that did not alert the body's defenses. And this discovery served as the catalyst for the vaccine race of 2020.
The parallels are stunning. The smallpox epidemic in London in 1721 broke out at exactly the right time, allowing Lady Montagu to chart the course for adoption of variolation. It seems like the COVID pandemic also broke out at possibly the most fortunate time for humanity. Had it broken out earlier, we would have not had vaccines being developed at such a rapid pace.
Definitely. In fact, the two companies at the forefront of mRNA vaccine development, Moderna and BioNTech, which produces the vaccine in collaboration with Pfizer, are very young companies, and they have very interesting stories. In 1990, Ur Shahin and Özlem Turesi are physicians who met while working at a German hospital, and they fell in love. The couple's primary area of interest was cancer immunotherapy, and they co-founded BioNTech in 2008, aimed at developing cancer vaccines based on mRNA technology. And in 2013, they even hired Carico as their senior VP, which showed their faith in the technology's potential. Meanwhile, Derek Rossi was a postdoctoral fellow in stem cell biology at Stanford when he first read Carico's papers in 2005. He found that mRNA held the key to another monumental game changer, stem cell therapy. And in the summer of 2010, Rossi presented his research to the CEO of the venture capitalist firm Flagship Ventures. And a few months later, Moderna was born. All the research on mRNA that these companies were working on over the last decade came to a head in 2020, just at the right time, and enabled them to design a vaccine within days. mRNA has proven to be the most fruitful vaccine technology with both shots at over 90% efficacy, which is much better than what traditional vaccines do.
What does the future look like for mRNA technology and vaccine development?
The success story with COVID has brought mRNA technology into the spotlight. It's the new kid on the block with the fanciest boots and the freshest clothes, and funding is no longer an obstacle for its advancement. And everybody wants to get in on the act. Cash is king, after all. And researchers are now looking at potential vaccines for malaria, HIV, herpes, etc. BioNTech's mRNA cancer vaccine has just started its phase 2 trials. They've also published data on novel mRNA vaccine approaches to treat autoimmune diseases. Another area of potential application is gene therapy. As it stands right now, gene therapy is very complex and an extremely expensive process, and it's largely inaccessible to the general populace. Scientists believe that mRNA holds the key to inexpensive gene therapy to treat ailments like sickle cell, inherited liver disease, just to name a few. A true revolution in vaccine manufacturing is upon us. And mRNA tech might be the answer to many of the diseases that have plagued humanity for centuries. It's safe to say that this just isn't the future of vaccines, but of pharmacology as we know it.
Scientific progress is a funny thing. For millennia, humankind has been a sitting duck to a variety of diseases. Diseases that cause disability, diseases which cause death and the fall of great leaders and civilizations. But in the last 3 centuries, we as a species have risen. We've created new techniques to protect ourselves. Maybe one day we can evolve into a species that's resistant to all diseases. But until then, let's be thankful we've got some brilliant minds and some great vaccines on our side. Key sources for this episode include Angel of Death, The Story of Smallpox by Gareth Williams, Antonio Regalado's article, The Next Act for Messenger RNA Could Be Bigger Than COVID Vaccines on MIT Technology Review. For an exhaustive list of sources, please visit anchor.fm/Pursuit of Prosperity. Pursuit of Prosperity is a fortnightly podcast written and produced by Shubham Vyas, Jishnu Chandar, Arnab Das, and me, Nikhil Nagaraj. Special thanks to Shubha Vyas. If you like the show, please remember to rate and review on Apple Podcasts.